The overall goal of this protocol is to characterize early brain developmental processes of human neural precursor cells in a rapid and reproducible fashion, and to define the effects of important extracellular factors in regulating these processes.
This method can help address important questions in the fields of neuropsychiatry and neurodevelopmental disorders, such as, do all affected individuals have the same cellular abnormality, or alternatively, does each display a person-specific dysfunction? The main advantage of our protocol is that it is rapid, highly reproducible, and easy to implement.
Though this method can be used to provide insight into neurodevelopmental disorders, it can also be used to study neurodegenerative and neuropsychiatric disorders such as Alzheimer's and depression.
Visual demonstration of some of these methods are necessary, as some techniques involve moving cells from one dish to another or require morphological analyses, which are best communicated through visual demonstration.
To lift the human neural precursor cells, aspirate the medium and wash the cells once with 1x phosphate-buffered saline. Aspirate the phosphate-buffered saline and add 500 microliters of 1x cell detachment solution to a well with the neural precursor cells. Then, incubate the plate at 37 degrees Celsius for 10 minutes. Next, use a P1000 pipette to add 500 microliters of phosphate-buffered saline maintained at room temperature to wash and remove the cells. Then, transfer the cells in phosphate-buffered saline into a 15 milliliter conical tube.